Yeah, many are still joining. I can see, but let's slowly get started. I think we have a quite tight schedule, so welcome to all to this workshop. Thank you very much for your participation, and thank you very much also to the presenters, which we'll see in a minute, for joining us. I'm Lars Dinesen from representing the Innovation Foundation from Denmark, and task lead on this task on a roadmap for implementation of novel monitoring methods, and together with Toge Høje, also representing the Innovation Foundation, and Cecile, and a working group within the partnership, we have arranged this workshop, and we are grateful for your participation. And I think on the next slide we will see who we are. We are too many, so for kind of a personal introduction, we will do that in the breakout groups in the second session. But fortunately, we have many wise persons, many experts, almost 60%, and we are altogether 60 participants. So
in your direction, so you can.
It looks promising. I think we have one which is not unmuted. And if we take the next slide, Cecilia, just this is the agenda of the workshop. You have all seen it before. We in a minute, Hilde, who is chair of the partnership, will give you a brief introduction to the partnership, and then we will start with the first session, which is 5 key presentations of about 10 minutes, and I'll be the ugly person making sure that we stick to the time. Then we will have a break about 10:30, and then we will have the breakout session starting 10:45 with the 4 thematic groups, and where you have already indicated which of the groups you think you belong to. And then we will have a wrap up, not a pretty short wrap up, and end 12:30 and be ready for lunch, I'm sure. So the aim of the workshop is to provide input for a roadmap for implementation of novel biodiversity monitoring methods. So there is a focus on uptake here, and of spreading these novel monitoring methods across Europe. And the roadmap will also guide future biodiversity work within the partnership. We already have a call on a research call on monitoring launched last year. And it will also promote to operationalize novel monitoring and identify research gaps, opportunities, and barriers. And we will build on the work already undertaken by Europa Bonn. So we are not inventing the wheel, but trying to build on what is already there. And one key thing will be to look for opportunities for standardization and for harmonization of approaches, and the work will also feed into future activities within the partnership. For instance, there will be capacity building events in the future within this same task. Next slide, please. So this is what we're aiming for, having input for this roadmap, and you will, as expert, have the opportunity to provide feedback, because we'll be a small core group writing up a draft workshop report draft, and that will be circulated to all of you, and you will provide have the opportunity to provide your input to that draft report before we finalize it. So I think with these words, I hope we will have a great day, and Hilde, chair of the partnership, will give you a brief introduction to the partnership itself. Thank you.
Thank you, Lars. I will be very brief because I will try to do it like in a nutshell, maybe 5 minutes. But with Biodiversa Plus, it's a network of both what we call research policy actors. So we have research ministries and funding agencies and foundations on board, but we also have ministries of environment and environmental protection agencies. So it's a network of these type of partners. It's a network co-funded by the European Commission. So it's also from the DG Research and DG Environment as part of the European biodiversity strategy. So we are part of the strategy to make the link between science, policy and practice. Biodiversa Plus is not an entirely new network because we are building on an existing network, which was Biodiversa. Biodiversa was running from 2008 until 2021. So we are building on what we were doing in the past, but we have significantly expanded the portfolio of activities, and also we have expanded the number of partners and the number of countries that we cover. So we currently cover around 37 countries, from the north to the south and from the west to the east of Europe. And next slide, please. So in terms of our activities, I will not go into all the details of that graph, but this is just to show that we have several working streams that are closely aligned with the objectives of the European Biodiversity Strategy 2030, but also now with the new post-2020 global biodiversity framework. So whatever we do is strongly entered in this new policy context. Next slide, please. So in terms of the type of activities that we currently cover with the network, we have on the one hand, we have the focus on the supporting and promotion of biodiversity research. And that is something that we have done in the past with Biodiversa, but now we are upscaling this. So every year we launch a call for research proposals with an amplitude of around 40 million euros, each time focusing on a certain topic. And then when these projects are funded, then we also try to support this project throughout the lifetime, also in terms of bringing the science to the policy level and also helping them with capacity building activities, stakeholder engagement activities, and also supporting them with open science. The second part of the work, and that is also the part that is of most relevant to today, is a strong focus of Biodiversa Plus on biodiversity monitoring. And here the ultimate aim would be to set up these transnational networks of harmonized biodiversity monitoring schemes, building on existing national and subnational schemes. So really building capacity for harmonization and also creating like this exchange of best practices across borders. And that is, of course, something that we do not do alone. We indeed do it together with Europabon, with the European Environment Agency, with the GRC, with the Knowledge Center, and with many other partners in the field. So it's really a joint and collective effort with the overarching aim to support biodiversity monitoring activities and to really have much better mobilization of data also for policy making and implementation. Then we have also a focus specifically on knowledge for nature-based solutions and engagement with the private sector. Then a big stream of the work that we do is focusing on science policy interfacing, so bringing the science to the policy level, but also better connection between research, monitoring, and policy. And then also we have a stream of work focusing on the global action of the EU. And that is also the work that we do in relation, for example, with the Convention on Biological Diversity, but also with the IPBES, the counterpart of the better known IPCC, and also the work that we do together with global research infrastructures like GBIF. So this is a bit of the portfolio of activities that we do. But today's workshop is, of course, in the context of the biodiversity monitoring activities. And that's also quite a broad set of activities on itself. So it focuses on harmonization of protocols, methods, data interoperability. We have also indeed this focus on new tools and methods for biodiversity monitoring, but it also goes all the way down to better use of monitoring data, both by research for like trend estimation and scenario building, but also better use of monitoring data by private and public policy making. So that's quite encompassing. Next slide. I think I'm done for the introduction. So this is a bit the Biodiversa Plus in a nutshell, but if you would like to have more information, you can always reach out to us bilaterally. And we also now have a brand new website where you can find all the information on our activities and also on the type of products that we produce. So with here, I will leave it here and I will hand over back to Toke.
So thanks very much, Hilde, and here we have the program of the first session of today, this morning. These five exciting keynote presentations, and the first will be by Tia Cho from Europa Bonn, and she was a lead author of the report from Europa Bonn held last year on novel monitoring technologies. So please, Tia Cho.
Hi, everyone. Good morning. Thanks, Lars and Hilde and Toke for having me. Just need a minute to share my screen, if you'll bear with me. Okay, everyone seeing the slides properly?
Yep.
Okay, I'm seeing nods.
Yep.
Okay, so as Lars said, I was part of the task in Europa Bonn on assessing novel biodiversity monitoring technologies and methods. And without having to say it, the title speaks for itself. It has quite a lot of overlaps with what Biodiversa Plus is doing here today. And so we're really excited on how you will be building on our current work. Just to introduce Europa Bonn for those of you who haven't heard of it before, I know some of you here in attendance actually came to some of our workshops, so you'll be familiar with it. But Europa Bonn stands for Europe Biodiversity Observation Network. And the overarching project is aiming to design a biodiversity monitoring strategy that works and is fit for purpose for Europe and can be used so that data collection to data integration, and then at the final end of the pipeline, it's integration into policy. All of that is a co-design. So we're involving our stakeholders and policy users of that data so that this entire pipeline of biodiversity monitoring works and can be used and operationalized, as we say. And I'm part of a small task in this giant project.
I think the slides are not advancing, just so you know.
It's okay. I'm just introducing at the title. Sorry. I'm part of the task 4.2 with Rob Patchett and led by Maria Dornelis here at St. Andrews. So it's a little bit squished. And so these are our aims, where essentially our work was to identify and systemically characterize any novel technologies or components of technologies that could help us monitor biodiversity better for Europe. And second, to assess whether these are ready for implementation. And then lastly, identify areas where monitoring is not appropriate to use novel methods. Sorry, I don't know why everything's so squished. So our task workflow initially was to start with a search and compilation in literature, gray literature, makerspace or startup websites, just to get a broad internet view of what is being developed and what has been developed in past years, but not, let's say, developed but not put into implementation or not used widely. So we came up with this metadatabase, and then to do the assessment, because I'm a coral reef ecologist. I dabble in macroecology, but I'm by no means an expert in all of these fields. So we conducted a workshop where we invited experts similar to this workshop to discuss the assessment of these methods according to taxa and method classes. And then some of you may have seen it, our review on the findings and assessment for Europabon. For our meta database, the initial search, it is basically a very large spreadsheet on metadata of methods. So that's why we call it a meta database. And essentially for every method or method component that we found, we basically want to understand, first of all, its developmental stage, whether it's very nascent and in its early beta testing, or whether it's something that's been published and potentially ready for implementation. If possible, the estimated cost, because that's something that Europabon is interested in, is how to improve the cost efficiency of biodiversity monitoring, which hopefully novel methods can alleviate, but some of them might increase the costs. And then also the EBV classes. So EBV stands for Essential Biodiversity Variables. It's a group of metrics that Henrique Pereira at IDIV has worked on over the past few years. He's the lead for Europabon. And these are essentially metrics that we want to monitor for that cover everything from the population to the ecosystem level. So indicators that we want to track in our monitoring. And then other fields on just the input data required, output data required, temporal and spatial resolution, as well as scope. And I'll just bring up an example of that metadatabase so that you can all see. It's not public yet because our task is still in the process of being finalized. But this will be public once that is. And so you can see we have lots of different technology names. I think our total tally is somewhere in the 300s now. And we've attempted to fill in the metadata as best as possible. But for a lot of these methods, because they may be components, so they're not full methods that generate from sampling to generating a biodiversity metric in the end. They may be just a piece of technology like data storage or data transfer that helps part of the monitoring data collection process. So we do have a lot of gaps in this metadata, but that was a really encouraging first step in our task. And I now have 10 minutes, so I'm going to fly through our initial summary and methods, but if anyone wants to ask me questions on slides furthermore later on, please feel free to. So just as a summary of what we saw preliminarily, a lot of the novel methods focused on remote sensing, AI processing, as well as technologies or methods that just help integrate data and synthesize them for better conclusions and trends to be observed across a larger region. And so we held a workshop to improve on this initial finding to assess whether all of these methods, these novel technologies that we found, are suitable and ready to be implemented. And they share quite similar objectives with our task. I'm not going to go into detail on all of these, but I just want to highlight that we asked our experts to use a technology readiness scale developed by NASA for their prototyping of space exploration technologies. We adapted this for our methods. So it breaks down what we started with the development stage of developing, testing, ready, to this 9-step qualitative scale. where it spans from basic method principles observed all the way to an actual method proven in their operational environment. And so we split all of these experts into breakout sessions sorted by taxa and method classes on the different days. And we asked them to just off the top of their head list all of these new technologies that they know of and assess whether they were appropriate or not and grade them according to this scale. So preliminarily, we found that our experts provided a lot of methods on data collection, and some of them relate to analysis and data integration, but on the most part is mostly about collection because that is the hardest part to improve on. And again, like our preliminary findings, they were focused a lot on remote sensing and AI. But also a lot of the target monitoring metrics focus on species distributions and abundances. So these are the EBV classes I mentioned earlier. They're split into these broad categories from species traits, populations, all the way to community composition and ecosystem structure. And within each of these categories, we have the indicators. So you can see a lot of the metrics relating to population genetic diversity or effective population size, or things such as interaction diversity. These are not as well focused on with the novel methods. A large majority of the methods focus on understanding species distributions, abundances, as well as some traits such as phenology, movement, and morphology. So it's a saturated and well-studied field, but it's quite patchy and skewed towards a few metrics. And most method classes had some implementations that were definitely ready for more widespread deployments. So over here, you can see I've taken all of the methods that our experts provided, and I've grouped them according to method classes, so broad categories. And then here's a distribution of each of their technology readiness levels. And the distribution ranges show that the readiness is spread quite widely for most categories, but you do see a lot of them have like at least one or two in the maximum, definitely ready for implementation stages. And it's just a really broad spectrum, but that for most of these categories, there is something that is applicable. And when I split this into taxa, so the same methods, but grouped by taxa, we see sort of clearer patterns, maybe not as widely ready as the method classes would show. But we see that some taxa are definitely lacking in the novel method developments, especially in algae and aquatic invertebrates. Just to remind everyone of the scale, actually, in ecology, implementation might not actually require methods to be at 9. So for example, I myself have used methods for fish when they were somewhere between 5 or 6. And so when we look here, actually, a lot of methods are within that range and potentially ready. And so I've gone a little over time, but just to touch on some overarching themes that our experts pointed out, not just specifically for one method, but across the board, there is a need for proper standardization. As this workshop is here today, and it's still a huge need that needs to be addressed because it needs to be interoperable between our member states, and it also prevents misuse of data. With synthesis and ecology, if we don't have the standardization, you're comparing apples with oranges. But also, when we're using new methods and technologies, sometimes that increases the need for data storage and infrastructure, especially if we're scaling them to the level of Europe as a region, not just within nations themselves. And so we need to also pay attention to that if we're developing networks. Say sensor networks would require a lot more infrastructure than deploying human observers. And finally, there are lots of gaps in reference knowledge and databases. So there's still need to clarify taxonomy and or having the proper databases to collate all of this data together. Yeah, so just going to end my talk here and allow time for questions. So validation is still a big part of the work in implementing technologies. It's shown for some model systems, but the work is still needed in order to invest in the testing and validation of methods for other taxa and or in other environments. And longevity is a reason not to change, but also because we'll require initial investments to put all of these novel methods in place. So thank you. That's me.
Thanks a lot, Chair. Thanks very much, and thanks also of being alert of time. I think we have time for one question before we will proceed, and of course, thanks a lot also for enlightening us of what happened last year at your workshop, and so we can build upon these results in this workshop here. So, Ola, I can see you have raised your hand for please a brief question and a brief answer before we proceed.
Yes, I want to ask, there were no studies regarding fungi.
Sorry, fungi?
Yeah, you didn't list as regards taxa, you didn't list that, I think. Or was it included in plants? Or?
Yeah, I grouped them under plants because it was for the practical, let's say like the practical organization of the participants, so that we didn't have one or two experts alone. But we did include them under terrestrial plants.
Okay, thank you.
Mm-hmm.
Okay. Thank you. We will then proceed to the next presentation, and that is Elaine talking about and presenting on ARISE, a very ambitious project and a country case, I think, from the Netherlands. So please, Elaine.
Morning, everyone. And luckily, I'm not alone. I see also Dan is here from ARISE. So if you have any technical questions, I divert to Dan. All right, let me put in presentation mode. So ARISE, yeah, it's an acronym, obviously, with many of these scientific names. And ARISE is a large national infrastructure. But so there's other people also involved in the more research realm. So Dan is involved, for instance, with Mambo, and Daniel Kissling is probably a known name through the Europabon people, who's also connected to ARISE directly. It's an 18 million research infrastructure project. So this means that we are responsible for creating the national infrastructure for all ecologists, biologists, computer scientists that are involved with species recognition. Ultimate goal is obviously to know exactly what's out there. The biggest problem, we are all aware, is that we tend to not know. So what methodologies can and can we not use? So we're not really research oriented. It's really an infrastructure. And our main focus is on the one hand on the DNA infrastructure, which I don't dive in too much today. But for instance, here we are also connected to the Biogenomics Europe initiative, where we really feel that the environmental DNA will be the solution to finding biodiversity. And for that, we need a reference database for all known species. Starting with the Netherlands, but BGE will help us, of course, on the European level. At the same time, we are shifting towards the sensor technology, which is more relevant for today. And one of the cameras that are, for instance, tested in the field is the Diopsis insect camera. But we're also connected, of course, with other cameras like wildlife monitoring cameras, sound recognition, radar technology, all sensors that could be deployed automatically or autonomously in the field with solar power, 4G connections, you name it. It's basically the idea that we set up an infrastructure to connect to these devices. regardless of what they are or who built them, and creating the standards for them. It also means that we need a lot around digital services. Well, Dan can tell you how many terabytes of information will be picked up by a sound recorder. It's massive. So not just a data storage problem, but also a recognition problem for which we want to provide the platform. So we're not going to build a recognition engine with just a model, but basically what we've done is create a platform where multiple algorithms can be stored in an algorithm repository. We have now provided the automatic deployment, so the model can be clicked on and chosen and then run for your analysis. And for that, we connected to the National High Performance Computer. Now, this is all still in prototype, we're testing, but the idea is to scale this out. And even though our assignment is for the Netherlands, we'd actually like to think about European or even global scale when necessary. So scaling up to data always in the back, or actually the front of our minds. Now you can imagine that it also requires connecting lots of different data, from metadata captured in the field about your location, time, who's been sampling or which sensor has been deployed, all the way to analysis, what analysis was done, which algorithm was used, and all of that can be merged together in this connected data fields, knowledge graph, whatever is actually at that point the standard. But for this, of course, we first need to have a proper data management system. And this is our biggest hurdle. So we're investing a lot of time in this. It needs to be fair. So there needs to be a registration with persistent identifiers. And all of this is now being developed. And as soon as we have it, we think we can deploy this also to all the data sets that we basically want to connect. So also outside of Arise, right? We're not collecting just our own data. The idea is that we kind of set the standard and allow connection of known databases for analysis. So some of the, just a few impressions. So we really want to create this online platform, be it the Spotify of biodiversity data, at least on a national level, where you come in and Arise provides you digital tools to get connected, whether that's sort of your intake to the laboratory facilities because you want to do eDNA monitoring, or you want to connect your sensor, or you already have sensor data and you just want to use the digital species identification environment, or you have a very neat additional tool, which is a widget and can be deployed on your own website. All of these should become available through the Arise platform. So a few impressions, for instance, on the DNA side, we've now created what we call a target list. So remember that we want to have a DNA barcode for every known species in the Netherlands. And we did a gap analysis to see what do we have, what do we not have. This is now sort of an interactive website where you have a search on a taxonomic level to find out if that species already has a barcode, yes or no. So we've chosen to actually take something that used to be a, well, Git repository table and quite the overwhelming information and put it into a more user-friendly environment. And this is also one of the, I think, exceptions that Arise tries to achieve is really make it user-friendly or user-interactive, because we feel then we can reach actually a larger audience. So this is, for instance, in the background, it still ends up in a table, but instead of starting all the way at Animalia in this huge table with all the metadata, once you dive into a particular species, the columns actually reduce. So it becomes a bit more insightful. We've also just have what we call a minimal viable product out in the field for testing with a bunch of users that helps us to capture metadata in the field. Nothing really new, but we discovered that actually collecting a specimen in the field, even if it's a single species or a bulk sample, registration with, for instance, collection registration numbers wasn't available. So you could register your observation, but you couldn't really automatically attach a collection registration number. So this is what we've been doing. So every new sample that comes in gets a collection registration number. It captures your location, time, date, and everything that needs to be there. What is also included is, remember, that target list. So the little heart shape with wish list is basically our target list indicating if this species has already a barcode, yes or no. So to also really encourage our specialists in the field to go and find the missing species. And this is our digital species identification environment. I'm not going to give a full demo. I think you can ask Dan for this. But here are the cases that you come in with your own data, you pick your sensor. So for instance, we've done this for the Diopsis camera. You can browse your images because there will be empty pictures or there will be a particular time lapse that you want to analyze. So you create your data set, you pick the algorithm that fits. Right now, you can only pick one algorithm, but in the future, you can pick multiple algorithms to also do an evaluation between algorithms. You analyze and the results will be stored with your original media files. So this is currently, I think, in test phase for Diopsis. The idea is to add on for various types of images, sound, radar. In the future, there will be also training. So we hope to create on the platform a training module so you can train your own algorithm and a whole annotation module so that everyone can actually help annotate and sort of give this feedback loop so that the algorithms can also improve. Now, last one. Also in the field, we've noticed that there's a need, for instance, to insights in your sensors. In this case, the ARISE monitoring demonstration sites team, it's a mouthful, but they're basically testing various sensors in the field. Right now, I think they tested about 7 or 8, but there was not one overview for all of these sensors. Some were on SD cards, some were with 4G connections and landed on an FTP server. They've now created this virtual pipeline, bringing together all of that data. In this case, what you see is, for instance, a wildlife camera, and you can easily go in here, go to the data viewer, pick up all of the images, see which are the latest or they're empty pictures, but also check on the battery status or see if the solar panel is down or your SD card is full. So just a couple of these services, and it's all in this philosophy of building this core infrastructure. So bringing together both a core data management system, including also the access control, which you are developing this year, API services, so that you have end user services that you can use through the ARISE platform. And also on the other hand, you see that there is a connection to be made with the known infrastructures, both national and international. So ultimately, this is the goal, right? We want to have this species reference database so that for any species, we have all the information at the tip of our fingers, and you can ask it questions because it's all interconnected through the persistent identifier infrastructure and in a fair way. On the data management side, maybe for those that are interested, because this is the hardest part, we've chosen for quite new technology here. Some of you may remember 10 years ago, we would have the data warehouse where nice tables were kind of sitting and they were updated maybe on a monthly basis. Then we wait for, no, let's do it unstructured. And there's just a lake, a data lake, and you can pick up whichever data set that you want or dump it in it. Right now, we're choosing sort of for a combination of the two, where you have nice data sets, but also maybe unstructured data, and you pick it up and you make sure that you have a harmonization layer so it can actually communicate to each other. And that allows that you have very different types of data connected and you can put services on top. Now, again, for details, please contact my colleague, Chantal Huibers, who couldn't make it today because she's celebrating in Panama. But I think this community knows her well through the Easy Rider project as well. Right, so for today, for this year, some of the things that we really need to focus on, like I mentioned, it's identity and access management, also the taxonomic backbone. It's kind of the pink elephant in the room. On a national level, we kind of know, but on the European level, it becomes already a bit more sketchy, let alone on the global level. So I think our community also really needs to connect on this topic. What do we choose? What do we not choose? What's the best solution, knowing that it will not be perfect ever? Focus on how we add persistent identifiers, because this is a really important step in the FAIR infrastructure. And sort of data strategies. Our previous share already in a previous presentation mentioned, lots of data needs to be stored, and there's no place to do this. So who should take the responsibility to do this? ARISE in the beginning basically said, like, nope, we're not going to store any data. We'll just pick up the data wherever we need it. But then, having the experience of deploying sensors in the field, we noticed there is not one place to store it. There are some data standards, but not sort of, there's data standards for the wildlife cameras, there's some for the Diopsis cameras. So how do we actually merge that together? And maybe should we even think about what if we did allow to store all data? Maybe we should collaborate with the national institutes and say, hey, let's work on that data storage, and say, if you do not store it yourself, or there's not an API to connect to, here you go, put it here, make sure that you adhere to these standards, and then you can connect to our platform. And that's it. So lots of ambitions. We're now in year 3 out of 10. So we have a 10-year funding and probably another round as well. So we're here to stay, and we'd love to collaborate and hear more also from your experience, what we should and should not do.
Many thanks, Elaine. Very, very interesting and very, very ambitious as well. I think we have room for a few questions. I have one. How do you see the prospects for replicating in other countries if you succeed with this ambitious project?
Yeah, so it depends what the country already has, probably. So I mean, the technology doesn't care about location, really. I think the biggest hurdle might be the taxonomic backbone, but the rest of the technology is actually quite scalable. So depending on where the country is, there are some countries that don't have anything, so they're immediately interested. Well, that looks great. How much would it cost me to deploy it in Morocco? And I'm like, well, wait for it. Other countries may already have particular components, so then you have to figure out how it fits in. I hope that we build it in a modular fashion so that you can pick up the bits that you want and have your own where you already have something.
Thank you. And do we have further questions? I see Florian, you have a question, please.
Yes, thanks, Elaine. Really, really good. So I really see this happening at a couple of countries, that the pull effect, that some go ahead and show that it's possible, is more leading to a transformation than trying to solve all problems at once. So I think this focus on national solutions seems to be reasonable. But concrete questions regarding taxonomic backbone, because this is really, as you said, what did you say, pink elephant, whatever. What is your pragmatic approach? Which taxonomic backbone do you use? GBIF or yeah, what are you linked to?
Yeah, so no, we've chosen to start with our own national taxonomic backbone. So we have a register for all the species in the Netherlands that are known, which is maintained by Naturalis. And then we send it as in the checklist bank of Catalog of Life. So it does pick up sort of the additional things that it could pick up, and from there we take it. Okay, yeah, but I 100% agree. We need to talk about this issue sooner or later. Yes. How to scale. Yeah, no, it's maybe a bit unfair, but there's also bits for Arise that is not. It's my problem, but I'm not sure I can solve it, because of course this has been an ongoing discussion. So.
Okay. Thanks. Thanks very much, Elaine, for your very interesting presentation. And Florian, actually, I think you are the next presenter, presenting on standards for molecular biodiversity assessments in freshwater ecosystems as a case. So please, Florian.
Yes. So just to let you know, we are still victim of a cyber attack. I'm connected via my mobile phone. In case connection interrupts, I will be back a minute later again, hopefully. So just to share my presentation. So I hope, do you see the correct screen? Yeah. Or is it?
Yeah, it's fine. We see it fine.
Okay. Yeah. So I led a cost action with which included almost 600 participants, depending on how you count it, and over 49 nations were formally involved in this cost action, which tried to think about ways to include DNA-based monitoring in existing international water biomonitoring frameworks, specifically the water framework directives. This talk has 3 propositions, and I will use them to walk through. So the first proposition basically is we need biodiversity weather stations, and eDNA methods can help here a lot. Oops. Sorry, my slides are jumping. So we all agree on that. We have too few data on where and how biodiversity changes. And what we know from environmental DNA assessments or DNA-based assessments of bulk samples, that we gain with very little quite a lot. This is an example which I like to show. It's from Didier Pont and his group. Looks like I have automated slides moving forward, so apologies for that. So what you see is, this is what you get with an eDNA sample in the River Rhone from 1 year compared to 10 years of electrofishing. So basically, with eDNA, you integrate much better the species diversity at a certain site. But of course, all of you know, it's not a perfect method. It has errors, false positives, false negatives. Abundance will remain an issue. But what we see also beyond water, we can use environmental DNA, for example, to monitor organisms living in the canopies just by collecting rainwater that flows through the canopy and then sequencing it. And I see also that Christina is here. So the recent method advancements using DNA from the air in order to detect species living in the surrounding is a really promising tool, which is not very complicated. And the method is scalable and affordable. For example, we now analyzed much more than 4,000 malaise trap samples, and it just takes a couple of weeks, and the per sample costs are really low, so you can monitor a country. And we know that the methods are largely comparable to existing monitoring data, to traditional methods. But we have to say, you will never get the same. So if you sequence an organism or if you collect eDNA, you will always have differences compared to a sampling when you collect it via nets, because you get, for example, the gut contents or other sources of DNA material with a sample. So let me just, I always have to now press one slide forward and back in order to not make it jumping for whatever reason. What I want to show is DNA-based methods have been criticized because different labs use different methods yielding different results. And this is partly true, yes. But in DNAquanet, we performed method tests, and what we could show the diatom people, for example, using mock communities, artificial ones, river or lake true communities, sequencing these via reference labs and various test labs, you see that different labs can come to the same conclusions. So the method itself can deliver robust data. However, and that's also a result of DNAquanet soon to be published, there are strong methodological biases. We all know that the extraction method, the primer, etc., can lead to different results. But also, if you use really the same methods, the labs analyzing that can really come up with very different results. So this is really same bulk sample mix, 7 different labs. And here's the proportion of taxa found between DNA morphological reference and the DNA samples. And you see huge variation can exist. And this variation we need to talk about, because they won't just vanish if we believe in it, but we have to work on it. And it needs a formal structure, which means we need standards, how to actually apply the methods, and we need a quality management system. So how do we ensure that a measurement that was done is really correct? But the next proposition, is, if we don't do that, we are lost, at least in a legal monitoring framework, because we now have, for the Water Framework Directive, all across Europe, not only 300 ways to assess surface water quality, but we have 400 something. I would need to speak to Ecostat or the how many intercalibrated methods we have. There are many, and the intercalibration is never perfect. But if we now add DNA-based methods on top of these, there will not be 300, but there will be thousands of methods, and harmonizing results will be difficult. So we have to really act now. This is my key message. I might stop here. So we need standards and guidance. The revolution is here. We don't have to show that DNA methods provide useful results. We have to show how we can actually make them useful for national and transnational monitoring. But my last proposition, solutions exist. I think we don't have to reinvent the wheel. So if we look to forensics, if we look to food fraud detection, if we look to medicine, all the standards exist for various purposes. just speaking of corona detection, there are various different PCR kits in order to detect whether you're infected by coronavirus or not. And there's a scheme how to assess whether a certain corona test with a certain set of primers, you see various primers, is accurate and precise. And this is what we can also do with community assessments using DNA or eDNA-based methods. And that was on the stairway at home. My wife is a medical doctor. So in medicine, people are using newer technology, which still does not mean that the old diagnoses were wrong. It gets a bit better, but still the old standardized methods were right. So standardizing things does not mean that you have to stick at a certain point. No, the field moves forward and you can also move forward. So standards don't hinder innovation. They enable these actually, because we will have much better data. And this is what DNAquanet worked on quite a bit. So minimum requirements. What would be minimum requirements for DNA-based samplings? It's not about dictating primers or attack polymerase or filtered volume of water. No, it's about setting contours and providing recommendations which data or metadata also to provide. And that can be very useful. What we managed, we have a new working group at the European Standards Consortium at CEN, so the formal standardization body at European level, and Christian Meissner, the chair of a new working group at Technical Committee 230. That's the working group 28, DNA and eDNA analysis. And there are being standards developed. And this is where we also need community support. These standards need input and they need to be endorsed. So this is really something we have to work on, not only in the water sector. But at the moment, what I'm seeing, like what Elaine showed, at the moment, the motors are mostly pilots at national level, like eDNA Society with various projects going on there. Canada has the iTrack DNA project, a huge project with immense, amazingly good results and also standardization activities. But we also have the large international projects launching now. And just a couple of weeks ago, the eDNAqua plant project, which focus on eDNA libraries, including reference libraries for water-based assessment. So also this exists, and we need to bring these actors together and agree, then the future will be bright. Just speaking of the water level, so we have an existing legal framework. EcoStat is responsible for the context, how Water Framework Directive in Europe shall work, and for intercalibration. And ideally, what we should do is now we need a network to advise, so experts, a working group at European level, how things can be intercalibrated, then develop standards based on these recommendations that will translate to the national levels where analyses are being conducted. Unfortunately, this is not happening at the moment. There's no mandate group. I think this is what we really would need at European level, a mandate group at these respective levels. But so at the moment, the national groups are pushing that forward, but that's not enough. We need to think the next steps and we need to think internationally. So I think in the end, we need to move to ISO. There has just been a really good session, a whole day session on DNA-based standards at the United Nations General Assembly 77 Science Summit. And out of that, there came a key message, what's needed: minimum requirement standards for DNA-based methods, inclusive standards, and there needs to be funding, especially when translating things to the global south also. Yeah, in conclusion, DNA is not perfect, will never be perfect for monitoring, but it offers great chances to extend and scale up biodiversity monitoring. It's highly complementary and compatible to other methods. It's an option we should not miss. And with that, I'm at the end of my talk, and let's identify next steps from where we are right now. Thank you.
Thank you very much, Florian. And fortunately, we have a breakout group on molecular methods where some of your recommendations most probably can go in. you really have the feeling of this is a field rapidly developing, listening to your presentation, and also quite a really complex field as well, with great opportunities. I don't know if there's any questions to Florian before we proceed. I see Marcus, you have a question, please.
Yes, thank you very much. It's more a comment. I mean, there is a EU Commission, the Biodiversity Unit, which is now renamed to Nature something Nature. It has a different name. They have a working group on monitoring and assessment, and maybe it would be good to contact, in that case, Ann Teller, and see if you can have a link between the national. So this working group is Commission services plus member states, at least for the EU 27, you have an opportunity maybe to communicate this point and get some link to the policy side. Thank you. I can provide you with the contact if you're interested. Thank you.
Is it the Knowledge Center for Biodiversity
No.
you're referring to, or
No.
something different? No. Okay.
Yeah.
Because
No,
there's also
it's-
a monitoring.
Yes, there's also a monitoring subgroup, but I mean from the institution. So if you want to liaise with the member states also, one opportunity could be, and this of course has to be explored, is in fact the working group, which is linked to the Biodiversity 2030 strategy, bit following up from what has been happened in the last 10 years in the 2020 strategy. So, maybe there's an entry point, and yeah, I can.
Very, very good. Thanks for the remark, and happy to learn about the contact.
Yeah, and you can just contact me, and we can liaise with and see if this is a way forward. Thanks.
Yeah, thank you, Marcus. Any further questions, or as I'm not following the chat, I don't know if there's questions in the chat or comments in the chat. And if not, we will proceed. Thanks again, Florian, for your presentation. We will then go to the next presentation, which will be held by Adria and Charlotte, and it will be a kind of a national showcase of implementation on bats. So please, I'm not sure whether it's Charlotte or Adria who will present first.
I will start myself.
Yeah.
Thank you. Yeah. Hello, good morning, everyone. Well, first of all, thank you for the invitation to be here today. I'm going to share my screen first. 1 second. Can you please confirm that you can see the screen?
That's fine. Super.
Okay. Okay. Thank you very much. So yeah, good morning. Today we're going to present a very specific and practical case. So together with Charlotte, we are going to talk about how we use bioacoustics to monitor bats at European level. So most of you already know that bioacoustics is a great technology for monitoring bats because several reasons. It's passive, it's remote, automatic or semi-automatic. It's non-invasive and relatively economic and appropriate to monitor many habitats and different species altogether. But it's well known as well that there are some limitations. And this is something that have been mentioned this morning already in several talks. Lack of standardization, because it's novel and it's been growing quite fastly in the last decades. Basically, everybody use their own protocols and methods, and there's no guidelines, no specific protocols to monitor bats using bioacoustics. And also, it creates a lot of data to store, and there is no storage available like at the continental level. Because of that, I'm participating in the Eurobats Commission intersessional working group for surveillance and monitoring of European bat species. And basically one of our objectives is to update the guidelines that were published quite long ago. Basically in this book, there is no mention of acoustics, or barely a mention, about 2 sentences. And nowadays it is probably one of the main methods to monitor bats. So in the last 2 years, we've been compiling all the monitoring schemes that are taking place in Europe. We are doing that in an open database that you can find at this link in internet. And the objective is to put all the protocols together, try to spot common fields, common methods, and came up with a one single pan-European protocol to be used. And after that, I'm going to showcase the first example that we are going to talk today. We in Spain, where I work, we created like the bat monitoring program website, and the idea was to provide a useful tool, a platform for all bat researchers, scientists, volunteers, naturalists to accumulate the data in a standardized way and using the same protocols. And as you can see here, there are 4 different protocols. We call Cairo Habitats, Cairo Rivers, Cairo Boxes, and Cairo Roost. But today I'm going to talk only about the first one, which is the one using bioacoustics. The main important aspect of this database is that it provides one single place to store all data with the tiniest detail that bat researchers need. And there is no other platform that provides this opportunity. So basically, in terms of the acoustics, people, what they can do is just create the sampling localities with all the metadata required, pictures, geographical coordinates, altitudes, habitat, everything. Then they can send the acoustic data to a high-performance computer servers that we have in Barcelona, classify all these files automatically, and then validate them manually. And then they can have results, monitoring trends, proportions, habitat, species composition, all together in the website. This is, well, this is like for personal purposes, but also for institutions and organizations. And then thanks to all these data sets, what we're working on is just to create trends at long term, have species distribution modeling. There's plenty of opportunities to be done with this data. And also the second method that we're using it, and people used to forget about it, is how do we use bioacoustics in caves. To sample caves, historically, the best method have been basically going underground, tens or hundreds of meters, and then try to locate the bat colonies. But nowadays, rather than taking pictures, what we can do is placing an infrared camera outside and an acoustic detector, and then synchronize them to count the individuals that emerge from the cave and also identify them thanks to the acoustics. And using this method, it's been very effective, very cheap, and very useful for everybody. So we also open this platform to storage and to provide this kind of data. You can see, if you are not aware of this, you can see here in the top right how it looks like, bats flying from the cave, and then with acoustics, we basically identify the species. And thanks to this kind of methods, we can basically have a list of caves and then rank them according to their priority, the species sensitivity, vulnerability, conservation status, or any other criteria. And I'm going to pass the word to Charlotte to speak about the other case study that we want to highlight today.
Thanks, Adria. So I'm currently working on the project, the bat migration routes in Europe. The starting point of this project is the citizen science program Vigikiro in France. So you can see on the map the data that we already collected. So the observation was that a lot of people Europe-wide collect full night monitoring data. Yet we have very little knowledge about where migrating bats pass during their migration. And this is a key conservation point because those species are very threatened. So we decided to invite everybody with full night monitoring data to send them to us so that we can collect them. And could you please pass this next slide, please? So the idea is to produce distribution maps with those data and then use the hotspots of activity at different seasons. Next, please. To then connect the summer grounds with the winter grounds. Next, please. So with this project, I think it will be the first review of the most common uses of settings and machines in Europe. So not only from citizen science programs, but also from people that are individuals or professionals. And then my task is to define which possibilities we have to aggregate data from different machines and settings. And next, please. And so I would like to finish the presentation with the current needs of the community in Europe. So I was really interested in the presentation of Hélène because I think there would be an interesting connection with what we would like to do. So the program for the study of bat migration in Europe already started something in this direction to find secured and standard storage location. And we also have to deal with confidentiality management. And what we still need is a remote access for raw data processing so that anybody with a research program can access this database, but also people who would like to download processed data, and also a tool for a dynamic view of the results. Okay, so that is all from me. Thank you.
You're muted, Lars.
Thanks a lot. I was saying to both of you for these insights into the bat world, and certainly if we know a cave with bats, we will get connected to you. Do we have any questions to Adria or Charlotte? I see Petri, you have a question, please, or a comment.
Yeah, thanks for the presentation and also for the previous speakers. Very interesting topics, all of them. But my question for this very last slide for Charlotte was that you mentioned this European database of recordings of the bats. So have you been in contact with GBIF and discussing with GBIF colleagues, for instance, if they have this kind of plans already? I just know my colleagues from Finland, and they are closely developing a national FinBIF services for acoustic recordings as a main place for data storage. So is there something similar ongoing in other countries or on European level?
I was not in contact with GBIF, and actually I think the solution that Elaine suggested is really interesting because you have all of those national programs like the Spanish program, the French program that already have a secure database and a good organization, and if they can be connected somehow to an international platform, this would be really interesting. So I don't know if the GBIF is a good solution for this platform. Maybe I have to look into it.
Thanks. And there is a comment or question from Dan, please.
Yeah, thanks. It was really interesting to see this bat stuff. On the previous question, I'd be surprised if GBIF was intending to be the host for a lot of audio, because for existing media such as images, they mostly link outwards. So I think I go along with Charlotte's answer, but I wanted to ask about storage for bat audio. If there are some very large ultrasonic recordings, that presumably means more data. Are there any other special considerations for ultrasonic or bat recordings?
I'm not sure I understood the question correctly.
Bat recordings, do they have other needs that are different from ordinary audio, audible audio?
Well, the sample rate is higher for ultrasound, so it means bigger data. I don't know if Adria, you would like to complete the answer.
I think that the requirements that you're asking, it's basically the space that is available, the size, because handling this data is more or less the same as normal acoustic data sets. If we speak about algorithms to classify them or to handle them, it has to be like appropriate algorithms as well. But for managing and handling the files, it's an equivalent thing. The only problem is that most of the storage options that we have so far, they do not accept this kind of files. That's the thing.
Thanks.
And I think we have time for another comment question. Toke, please.
Yeah, thanks for a really nice set of presentations. If I understand correctly, then compared to the presentation by Elaine, your initiative seems to be like more bottom up and kind of developed from a specific user community that saw the need for data infrastructure and for data visualization tools. And I'm curious to know, how did you manage to coordinate this work, and how did you manage to fund it? Because Arise kind of came from one big funding, and you kind of maybe came more from just the need to have these infrastructure. And I can imagine that others might be thinking along similar lines. How do we make sure we develop such infrastructure? Can you elaborate a bit on this?
Maybe Adria, you would like to start, and I can finish the answer. So
yeah, okay. Yeah, I think that this question actually fits like both my and Charlotte's talk. So in my case, for example, that's completely true. So we start from the bottom, and then we try to scale up the project. We always like to think that if we start from the bottom, we just can provide solutions for the real problems, like talking to the people that are most needed. So what we created basically was this platform to solve two problems. That was the standardization and the storage. And this problem was common amongst bad researchers, not only from the Iberian Peninsula, but also from all around Europe. And we've been discussing that for a long time with many people. So basically what we did is we got the funding to create the platform at regional level or country level. And we did it, but always with the aim to be scalable. So not adding any limitations in terms of species, in terms of languages, in terms of whatever. So we tried to create a platform that could be used everywhere in Europe. And after that, I like to call it like the snowball. So as soon as we got the platform running, people from outside a region just like it, and they start using it. And that's the way we've been increasing the reach, how we reach the people. So many people every month is just adding data to our website from farther places. And the thing is that the advantage of using this approach is the platform, it's easier to be built, and then we can fix specific problems. But the platform is fully functional nowadays. So if you go from Finland and you want to provide data, it's ready. You can use it, and you can provide all the advantages. You have an organized database that you can download from with your old data. You can visualize your data in Finland, no problem. So everything's ready and it's finished. But that doesn't mean that the funding is finished. I mean, we still have funding every year to improve it, to fix problems, to provide. So what I'm doing now, like during the last years, is trying to connect with people outside and let them know that the tool is ready, it's functional, and they can be used at any point. That's the way, it's how we like to do the approach, rather than creating a big thing that then it's really difficult to be implemented at local level.
Thanks a lot, Adria. Charlotte, you wanted to add something?
Yeah, maybe the people from Spain had a bigger vision than we did, because we were really French focused at the beginning with the French citizen science program. So it was not possible to invite other countries because the language was not ready. So now this is where we are, and I think in the future we should work more with the Spanish people, so that the platform that is currently working can be friendly for international users. And for the European database, the funding that we have is really short, so we are always looking for new funding to continue the program.
So thanks a lot indeed for a very interesting example of practical implementation of bad monitoring. I think we need to proceed now and go to our last speaker, and this is Markus from the EEA, European Environmental Agency, and kind of providing the perspective of the agency on these novel monitoring. So Markus, please, the floor is yours.
Thanks a lot. Yeah, I have to admit I'm more on the learning curve here than really. I don't know what I can provide to this very complex question, except adding a few more layers on complexity here. And thanks to Cecile for managing my slides here. So this is a bit on reflecting from an institutional side on the biodiversity monitoring and all these efforts we see here in all these presentations, and maybe also further discussed in the sessions. So next slide, please. So our challenge, of course, as an organization, and I mean, we share this challenge also with many national organizations and agencies here, is the knowledge integration. So what we hear now today is mostly collecting the data and structuring it. So for us, then the next 3 steps, of course, are also important. So therefore, we, of course, have a very strong interest on harmonization, standardization, get the data comparable, get it traceable, everything which is also, let's say, more or less documented and written in the FAIR principle. To make use of it, and especially to make use of it in combination with other data. So next slide, please. From the knowledge dimension, of course, we have the data dimension. And here, this is just an example how we organize ourselves. So we used Inspire a little bit as one way to implement FAIR data principles. Of course, yeah, unfortunately, we've not fully succeeded here, but so we have to go further from there. But we also see that this very complex biodiversity monitoring is one aspect of this data dimension we have to integrate. To go to our assessments, where we look on ecosystems, we look on how this is, how they are under pressure, what are possible responses, and so on and so on. So here, I can just refer to the previous presentation from Elaine. She demonstrated also what the complexity also means simply to go to the accessible biodiversity monitoring, especially with the novel technologies. And we can play this game further and further, adding more and more layers. And unfortunately, or fortunately, there's also the need for adding complexity on these questions. So next slide, please. So one way how we look on that is, of course, we also try to structure this information. This is just an illustrative example. As I said, Elaine has illustrated it much better than I could do, because for us, the novel biodiversity monitoring is really new. So there are no, it's not really implemented in the current reporting lines. We see it now upcoming with the Water Framework Directive. We need the same, of course, for all the other directives. So that's just an illustrative example. And please, for the next one. So we have these challenges, and we see that these new methods, or in some cases not so new methods, really should help us to get a better a better information information room for this very important problem, because from the policy side, we have now 3-4 circles of biodiversity strategies, all with the aim to stop the loss of biodiversity, and it's still going on. Yeah. So far we had a failure in the policy over almost 3-4 decades. At the same time, we see also that the biodiversity monitoring was, and now I can say was, very underdeveloped in terms of information available. So here I refer a little bit on what Ian Donoghue from our scientific committee has also outlined once. It means we still have this limitation and bias in the biodiversity data in all aspects, and especially missing the abundance information. So here we see, of course, that these novel technologies can add a lot, and we are really keen on seeing that coming up also in the reporting lines, because currently the information is simply not fit for purpose for policymaking. So we make biased decisions. We see that a lot goes towards the charismatic species, also, of course, for the public awareness. But we are losing. It seems we are losing the baseline, especially the lower taxa, the taxas which are mostly lower in the food web, invertebrates. Of course, pollinators was a big issue here, but also algae, lichens, and so on. And the same seems to happen also for freshwater and marine. And at the same time, we need this aggregated indicator. So that means the data which is collected, provided, should be also be able to be comparable and be aggregated into indicators. And the policy always asks for one number. Of course, we always have to tell them, okay, there's no one number for biodiversity, but they look for aggregate information where they can simply communicate also changes. Yeah. So that means the data should also be fit for purpose for that aspect. I mean, I can only refer to this 1.5 Kelvin target from the climate change in the climate change negotiations. Yeah? I mean, the number is per se not correct because it neglects a lot of things, but it's a number they can negotiate on. And of course, adding all this complexity in the biodiversity as such, one number might be not necessarily the one we are targeting for. So I always said we need the appropriate complexity and the appropriate simplification. But well, designing monitoring sites, of course, should keep in mind that there is a need for the further use of this information. And this is the key message here. And then, and this is a problem we see now partly addressed or even fully addressed in some of the on national level, like the ARISE program, is we have an institutional gap for gathering, compiling, and hosting data. Means even with the services they already exist now, like the Copernicus remote sensing based services, we have a problem to have the capacities to assess this information. And this is something which developed in the last years, simply with now we are more and more getting the data we need. And I see the same now in the biodiversity monitoring. We are asking for this information for many years. Now it's there, but now we need to develop the capacities to analyze and assess and link this information, of course, with the other information to get the knowledge structured and policy relevant. So there's an urgent need for more and better data, on the one hand, to enhance our knowledge base, but also the need for capacity building, especially on the EU level. And this also has been mentioned several times in all dimensions, in standardization, but also in the IT infrastructure, in hiring the respective, developing the respective capacities to use and assess this information. Next slide, please. So, what could be the way forward from our perspective, the institutional perspective? I wrote fair data, yes, because without fair data principles, there's no progress. Then forget it. Yeah, if you can't have fair data on board, we will not be able to make any progress. To be clear, therefore, this part of my presentation title was rather simple. So we see, of course, and this already has been nicely illustrated in the previous presentations, these novel technologies provide a lot of opportunities to improve, but they also create new challenges in handling this information. Yeah. And we see this not only in the biodiversity, we see this also in other sectors, as I mentioned, also in the remote sensing, but also in all the other information layers. So harmonized and standardized methods of monitoring, data storage architecture, also recalculation of time series, integration of existing monitoring schemes. This already has been mentioned. And again, I refer to the ARISE project here and the other initiatives to how to go forward there. What we need, of course, is robust change detection. So for the policy relevance, we need to not only to demonstrate what is the situation, but how the situation is changing over time. So single data point inventories doesn't help us a lot. They just give us some idea. Okay, there is biodiversity, but this is also something I see, of course, in these presentations already. addressed. So robust change detection is a key point here. And then, of course, the biodiversity monitoring design as such, what we need to make it policy relevant is the contextualization with the environmental conditions. Yeah, it's not sufficient to know, okay, there is this and this number of species, and hopefully also their abundance, but also what is the link to the environmental conditions? Yeah. So we just not only need, let's say, a biodiversity stations, yeah, like weather stations. We need the combination of the information. And I think the design of the monitoring, biodiversity monitoring, can already help in this aspect to take this into account and not just relying on other databases, but also see how this can already be linked and conceptualized in a very early stage. And this is all linked to this drivers, pressures, condition impacts circle. Yeah, we usually use. That means if you want to develop adequate responses, policy decisions, measures, you have to know what the pressures are, what is the condition of the system, how it is affected by the pressures, how to change that, how does this change over time, and then what is the functional trade between these environmental conditions and biodiversity, the species richness and abundance. So that means going more towards an ecosystem monitoring approach than just stick on the biodiversity as such and keep it there. Yeah, and then finally, yeah, I think as we luckily, and I'm very happy that we are invited to this, have now the Biodiversa Plus partnership. I really would think along the partnership dimension. And this is, I think, key to make this a success and to get the monitoring into the policy decision process. So the co-development with the respective institutions is key here. So it's not just handing over databases or platforms, products, websites, whatever, because you will hardly find the capacity to take that up on the institutional side. Yeah, and I could mention several examples here, so that there were platforms developed very nicely with all sorts of products and user interfaces and so on and so on. But at the end, there was no capacity to take it up from the other side. And especially now with this new dimension of computer capacity, machine learning, all this added complexities, which finally should give us the information we really need to make a difference in the policy lines. Ask for a strict co-development with the institutional partners. So we need this better link between research and policy implementation to make it a difference. So if you want to go beyond the research as such and link it to make it policy relevant, this is key, I think, in my understanding and from my experience. Yeah, and this is where I would open the discussion. I think that was my last slide. If you go for the next one, yeah, that's just something to look on. And now I'm happy to take up any questions. Thank you.
Thanks a lot, Marcus, and thanks for a very nice summary also for listing up all the opportunities and also some barriers and the link to EU reporting obligations and so on. Thanks a lot. We have not time, but let's take, I see Daniela, but please be very brief because we are now going into our coffee break, and I would also like that we introduce the breakout sessions before we go into that coffee break. So please, Daniela.
I will be very brief. I completely agree with Marcus's proposals, and I just want to remind you that, all of you, that biodiversity is huge, but the policy should go toward the monitoring methodology, minimum standards to be obligatory in member states, which is not the case. And also, it's not comparable in between member states per species based on a very low or uncomparable data quality. of monitoring methodology. So without that, any policy will fail. So this I support Marcus, and I support connecting environmental data with biodiversity data. Thank you very much.
Thanks, Daniela, for pointing that out. I think that's a huge challenge indeed. I think we will proceed. Thanks a lot for the 5 presentations. Really interesting. And thanks very much also for being so good at sticking to time, which is always a challenge and was really not a challenge in this session. So thanks indeed for all these insights. And now we will briefly introduce the 4 subgroups before we go into the coffee break, so you know what you are returning to in 15 minutes. So as you already know and have seen in your invitation, we have these 4 groups, breakout groups, not subgroups, but breakout groups listed, and you have already indicated which of the groups you would prefer to go into. So 1 on remote sensing, 1 on image-based approaches, group 3 on acoustics, and group 4 on molecular methods. And you can see the facilitators as well as there will be a support person as well for each of the 4 groups. So when you return from your coffee break, 10:50, you have a quarter. Please be in time, and then we will break out in these groups, and there will be an opportunity also to take a round table and introduce yourself. So please be back in 15 minutes for the breakout session. Discussions, that would be nice. So group 1 on remote sensing, where Petri was the facilitator. Are you able to present a few key points from your discussions there?
Yeah, let's see how it goes. We had a very good discussion and a lively group and a good different background. So with this Earth observation remote sensing, we of course were discussing different scales from satellites to LiDAR drones, and also we had an expert of radar data, which is a bit different. But anyway, all kind of technological approaches, how to use these for biodiversity monitoring. We were getting different wins as pros, which, for example, first one is that there's good political momentum at the moment to support this kind of implementation of new remote sensing based methods for biodiversity monitoring and detection. There's also funding available, at least for the next 2 years. So there's really a lot of demand and expectations for this kind of data. Then what it comes from the technical point of view is that the technology is quite major. There are a lot of good data products available, and with support of machine learning and artificial intelligence, it's possible to handle these data sets. Of course, the challenge might be that you need a big data facilities and the data cloud computing opportunities for that. But anyway, that's more or less available there. And then what it comes for the application side, how to use data. We especially highlighted that it's maybe not a silver bullet solution for all kind of habitat mapping or biodiversity monitoring, but remote sensing at different scales can provide very valuable supporting data. Maybe not so well fitting for the mapping itself, except for some particular habitats, which are maybe more open habitats or having a different lineations like Arctic or Myers peatlands, this kind of habitats. But especially when you have this starting situation of different habitat polygons available, you can use remote sensing for change detection in a quite smooth way. And also you can try to get benefit from the remote sensing when you are designing your management and actions for different conservation purposes. Then if you move to the anchor side for the missing or challenging elements, the one is that there's lack of in situ data that is really much needed to support use of remote sensing, and validation data is one part of that. Then there's maybe need for continuing this kind of opportunities like we have today, this workshop where different expertises come together and discuss and start to learn each other's language and vocabularies. So we need this kind of culture for having multidisciplinary projects and experts to work together. What comes to the applications, there's definitely need for the data analysis and analytical capacities for the future. So that's maybe inhibiting at the moment a little bit of the use of the Earth observations for everyday use, but it shouldn't be too hard to reach this level. And then also there are some technical limitations still, like for example, cloud detection algorithms must be improved and that kind of, but they are rather minor issues in that sense. So the technology is there to start to be used. Then we discussed about what other actions to do and timelines and initiatives. It's quite a long list here, but maybe I can provide written comments later afterwards. But there was one example, for example, coming from this pollinator community. There's a spring project which is having a good policy-driven support, and then researchers implementing this kind of EU-wide pollinator monitoring scheme. So maybe learning from that experience might be something useful for remote sensing community also. But the challenge is that the pollinator group is based on available networks and a large number of expertise, while remote sensing needs maybe to have a bigger organizations involved and Copernicus and satellite data providers working together with those who are able to handle the data and make the analysis together. But anyway, it's a good example. Yeah, and this mixed expertise for collaboration, it was highlighted. It was mentioned already earlier, but that's very important. We need to improve this in situ data collection somehow, but we didn't have really time to discuss that. Who should take care of that and what kind of timeline there should be? Who should be leading this kind of process over to Europe? But where we can learn, we can, for example, focus for Corinne land cover process, how the data is harmonized there and what kind of data collection is done, and maybe building on that experience, it could be possible to also focus for habitat monitoring. And as always, there's uncertainties are important, and for example, yeah,
flag the time, Peter, and just
this is very last two very last points.
Super. And we have the
so
board
with the initiatives who to contact when you are making, for example, uncertainty analysis for the maps. You can communicate with the local landowners or local amateur citizen scientists to involve them also for evaluating your data products. And then lack of money for field work, that was highlighted as quite often. It's a challenge. So I think I finish here. If anyone would like to update or comment something was missing. Otherwise, I will clear my notes and provide them to you so that you can catch up them. So
thanks a lot for this, and as I said, we will have a copy of your boards as well, so that will certainly go into the writing process. Then we have group 2. Please, Toge, you were facilitating, and I think PJ is wrap up for group 2.
Yeah, hi, so I'm PJ Stevenson. I'm chair of IUCN's Species Monitoring Specialist Group based at the University of Lausanne in Switzerland. Yeah, it was a discussion of image-based techniques, but to be honest, we stuck pretty much to camera trapping throughout this. I think, let me start with the wins. There are lots of wins. I mean, we felt that the tools and equipment for image-based techniques, especially camera trapping, are very well established. The technology is getting more easily accessible. Protocols exist for using it for research and monitoring. There are data sets out there. AI is improving all the time for image analysis, and there are several global databases that are collecting data. I mean, Wildlife Insights is collecting images and data. A lot of people's data are feeding into GBIF, and obviously national and regional efforts as well. Capacity building efforts are ongoing, and there are a lot of partnerships exist and collaborations moving it forward. And like the previous reporter, we also felt as a group that this sort of dialogue and this sort of discussion is very useful, and we were very appreciative of this effort. But what we felt then about camera trapping in particular, but maybe more generally, I mean, who should lead this work? That is kind of leading into the anchors, but who is the captain of this ship that can pull up the anchors and set sail and use the wind to move forward, if you like? You know, Biodiversa Plus, they're obviously, you guys are starting an initiative here, but where does EU environment fit in? Where does JRC fit in? Where do the national efforts fit into this? That needs to be clarified. Other main anchors for camera trapping, it's agreement. I mean, there are protocols, but what are the standards for collecting data and then managing the data? What are the standards for Europe and who should set those? Also, in terms of data sharing, how do we break out of our silos at national and regional level and improve data sharing and agree on suitable repositories for camera trap images? Also, we felt, and this may apply to other methods, but it's very important that there's a better understanding as well sometimes of the pros and cons of different methods. I mean, camera trapping is very useful, but when would it actually be more appropriate to use another method or just have people go out on the ground and count wildlife directly? Also, camera traps tend to just get used for species presence, but if used properly, they can get into issues of relative abundance and abundance. But again, bycatch data where non-target species are photographed or captured is not really analyzed as well as it could be. And we also felt, you know, one of the anchors, maybe we don't understand enough the main user challenges. I mean, we came up with some ideas, maybe other user challenges, some sort of survey, getting to understand what those challenges are for users, end users, would be good. And just overall then as well, we thought as part of that, better collaboration between some of the technical experts providing the technology, some of the database managers and some of the data end users would be good. And as I said, we felt that workshops like this definitely help with that. So I'll leave it there as hopefully an attempt at a summary.
Thanks a lot. Anything to add? Otherwise we will continue. Okay. Thanks a lot. A lot of questions and some solutions. Then we go to group 3 on the acoustics, and Hilde has left. She was facilitating. I don't know, is it Daniela reporting?
It's me. Yes, it's me.
Please.
So Daniela Hamidovic, Institute for Environment and Nature, Ministry of Economy and Sustainable Development from Croatia. So our small group was extremely brainstorming, and I will present you some of our discussion results. So basically, acoustic is the expanding field. It's evolved very quickly, and it has a evolving market for the affordable equipment. Also, it has a good research background, and there is a lot of very large amount of audio data incoming both from experts and from the citizens. There are some sound repositories available, and some citizen science programs are developed in some countries. So there is a direct benefit of results provided by powerful AI to small contributors. There is also acoustic indices allow analysis of a whole soundscape, which is evolving field. So there is the group of animals covered are mostly whales, bats, and birds, but some also groups are quickly evolving. So these were the wins. Now the anchors, of course, are data storage, data management, and data standardization. There is also the equipment anchor. So different sensors have different sensitivities. So this is difficult data integration and comparison. There is a lot of data missing from the databases regarding specifically the equipment, and the capacity and knowledge in end users is also missing. So missing is calibration data, maintenance, detection space. There is a lot of data variation, geographical, seasonal, etc. So there are a lot of dialects in different group of animals in different countries. Also, there are some international libraries, and some sound libraries are biased. There is missing field of data validation. So in in order to overcome these anchors and to, well, let the boat ship, the data storage needs funding. There is no need for a central repository, but different repositories should be able to communicate and to exchange data. There is also the funding and cooperation on open questions still developing in a scientific field. So these open questions, so it could be used by end users, should be funded in scientific field. Acoustic indices use in automatic ID is in development. It should be supported. The Biodiversa could help. So we said what it could do. It could make the international platform for knowledge exchange, and it could connect to Wildlabs Net Conservation Technology Network. So basically, we all technical experts in the acoustics fields in different group of animals could be connected on a Biodiversa platform in order to develop minimum essential standards for the usage of the equipment and for the metadata standardization.
Thank you. I think that was quite a strong recommendation. And our rush, we have only a few minutes left. We will try to stick to end at time as much as possible. So group 4 with Florian as the facilitator. I don't know who will present from your group.
I will present. Ola
will present.
Ola will present. Thanks.
As regards winds, we noted the big push from the political arena, from COP and the European Green Deal, the big need for affordable species information. And also we noted as another win that the methods have been increasingly cheaper, although still costs is a problem, especially perhaps for Southern European and Eastern European countries, perhaps. As regards to anchors, we identified 3 main blocks. One is about still lack of methods concerning sampling and extraction methods. And also when these have begun to develop, lack of standardization and protocols for these kind of methods. Also, it is a lack of awareness of policymakers of the potential of the methods. And the third anchor cluster is about lack of a taxonomic backbone, especially alpha taxonomy in many groups, and incomplete and sometimes low quality reference databases. And concerning actions, we think as regards lack of methods and standardization, it's important to create expert platforms, for example, cost actions for organized around different kind of medium, for example, soil, water, etc., where the problems may be specific in each case. And also use existing standardization bodies for this. As regards standardization, emphasis should be on minimum standards, so not hamper development of new technology, but there should be a minimum standards for different technologies. And then we need for tackle the lack of awareness of policymakers by creating networks, including them, and have a good communication strategy. Reference databases, it's both a need to identify gaps regarding taxonomic groups and which geographic areas within Europe that lacks good data for reference data, and also to check the quality problems of existing databases. And secondly, to support alpha taxonomy in badly known groups. Thank you.
Thanks a lot.
Maybe Florian will comment on something. No?
I think it was well covered. We need platforms that interact.
Yeah,
and the clear communication
I
strategy.
think it was quite clear. Okay, thanks a lot. We are going to the end. And Togo, do you feel for wrapping up at general terms before I'm going to thank for the participation?
Yeah, just very briefly, I think we're running a little bit behind, but I think it's fair to first of all thank all the speakers for excellent context setting of this discussion. I think we've had really valuable discussions in the breakout groups, partly because of those introductory speakers. So thank you very much for that. I also want to just repeat what was said, that we will be preparing a roadmap report out of the Biodiversa Plus working group. And we will circulate a draft for that, so you all have a chance to comment on our report. I learned a lot from this meeting, and I think one of the strong messages is that Biodiversa Plus could facilitate further capacity building in this field, both through online meetings like this, maybe even more specialized or even more cross-cutting. Now we were in our silos in the breakout groups, but maybe discussions about multimodal data integration could be as useful as well. I know that Europa Bonn intend to make their metadata base that Xie Xiao was speaking about available at some point. It's in a review at the moment with the advisory board, but I think that's also a useful context for these discussions, so we know what tools we're talking about. Yeah, I think a strong message out of this this workshop is also that we need to think beyond the technology readiness level scale that Xie Xiao introduced. What does it take to ensure uptake of these technologies? It's not just about the readiness level, but it's also about disseminating the potential and the applications for large-scale biodiversity monitoring. So yeah, that's all I had in a quick wrap up. I hope to see many of you soon again in continuing these discussions. Thank you, everyone.
Super. Yeah. Thanks a lot. Hope you had some good fun. And I also want to extend the thanks to the working group in the partnership for their support, and especially to Cecile for great support throughout this work. That has really been extremely helpful, Cecile. And you will hear from us, as has been said, so there'll be a draft circulated to all of you for further input. So hopefully this was just the start for something much larger. Okay, thank you and have a good day.